Hello and welcome to Behind the Lab. If you can just give me your name, the institution that you are at, and how you are affiliated with the MMPC.
Sure. I'm Brian DeBosch. I am a professor at Indiana University School of Medicine in the Department of Pediatrics, Molecular Metabolism Center. The way that I'm affiliated with the MMPC is that I have collaborated with Luis for probably 5 or more years in phenotyping our mice with multiple genetic lesions in the hepatocyte fasting pathway.
Thank you so much for that. With your affiliation with MMPC, have you done any notable work that you have used services from the MMPC that you would like to highlight, and any major grants and awards that you've won over the course of your tenure and any of the projects that you've done?
Yes. Let's see here. I have. We recently published in Cell Molecular Gastroenterology and Hepatology. We published work with Luis and the rest of the MMPC showing a structure-function relationship in how the urea cycle mediates whole body fat oxidation, or glucose and fat oxidation. We published that a little over a year ago, and in addition to that, Luis has been on how many grant submissions? Probably, I think 2, but there may be more. 2 grant submissions that we've made so far. We're still pending score on those, so no word yet, but I'm optimistic with their help.
That's so great. Are you working on anything right now within the metabolic disease area? Is there a specific project that you would like to kind of tell our audience about? And even if it isn't affiliated with MMPC, is there anything that you would like to kind of share on that end?
Yes, actually, this has to do with a collaborative grant that Luis and I have put in together on how a canonical circadian gene, period 1, regulates substrate selection and insulin sensitivity. And so the way that we've worked together is that Luis has kindly offered collaboration and services to perform euglycemic clamps on mice in which we made a genetic lesion in this circadian gene, period 1, in hepatocytes only. And so they'll be able to help us determine the degree to which the circadian metabolic regulator in the hepatocyte really mediates and drives substrate selection in the global sense in mice.
And does that research and does that information essentially are you guys going to end up publishing on this research, or is this going to be sent for usage with humans? What's kind of that process?
Yep. The hope is in the near future to publish those data in mice. We do have a translational aspect of the project that we hope to bring to human trial eventually. We're in sort of the proof of concept stages, but essentially what we've been able to do is sort of mimic the signaling aspects of period 1, mimic some of its signaling aspects, and package that into lipid nanoparticles to sort of really mimic the effects of period 1 overexpression and activation, which is what we've shown in our studies that are currently under submission.
Is there a service that you have utilized the most in your time at your university?
Oh, yes. Luis and the MMPC really are some of the very best at performing euglycemic clamp studies. I mean, not only are they so technically great at it, but they're also great collaborators, great advisors, and they really help start to finish with design of the proper experiment, proper powering, all the way through the technical aspects of doing and acquiring the data to analyzing it after the fact. They really are among the very best, and that's why we travel sort of far and wide to access being able to work with them.
This is so great. I know you mentioned or you touched on like the paper that you would be publishing in the future based on the research that you are doing currently, but is there any paper that you have done in your tenure that you would also like to highlight that's already been published?
Yes, do you mean specifically work that already has some of my work with Luis and the MMPC in there?
Or any paper that you've just been very proud of in your work?
Oh, yeah. Let's see here. Well, I described some of the work that I published on the period 1 story has been published. Most recently in Cell Reports, we just published in 2020. Was it last year? Yeah, I believe it was last year that we published some of those data in Cell Reports. But beyond that, my laboratory studies how fasting signals in the liver can actually modulate whole body substrate oxidation. And a related study was of a fasting-induced urea cycle enzyme called arginase 2. And we recently published that in 2024 in Cell Metabolism. We were able to show hierarchical control of the tricarboxylic acid cycle by modulating the urea cycle. And what that led to is a couple things. Number one, it showed potential new diagnostic biomarkers or prognostic biomarkers that can actually predict the onset of severe fatty liver disease in healthy patients by measuring elevated serum aspartate. That was one offshoot of the data. And the other is sort of the more or less novel regulation of the TCA cycle by urea cycle enzymes.
Thank you so much for sharing. I only have a few more questions. You already touched on it, just about the collaborative excellence that you've experienced with Louise and the MMPC, but if you could give me maybe two more things of what are two top things our audience should know about working with the MMPC or just your experience with the MMPC as a whole.
Oh gosh. Okay. I hope I don't sound redundant, but I really mean to drive home the point. Number one, their technical expertise is among the very best, especially in a very difficult and time-consuming, labor-intensive, just very experience-dependent assay like a euglycemic clamp. They're among the very first that I even think of when I'm thinking about doing that assay. But beyond that technical expertise, really, I mean, Louise is just a fantastic colleague. Not only is she nice and responsive and prompt and kind, but just a great advisor on helping you design the very best experiment right from the get-go, so that when you do the experiment at the very end, when you're interpreting the data, there are no regrets on, man, I should have done this, I should have powered it more, I should have added this extra control arm in. Just a really great advisor, collaborator, and scientist as well.
And I'm sure Louise would be honored just hearing you you share that information. So thank you so much for that. And my final question is, if you could give advice, or if you could even talk to your younger self at the start of your career, what advice would you give yourself, having known everything you've known in your entire career and your experience?
Wow. Let's see here. I think the best advice that I still tell myself today, you said tell my younger self. I'm old now, and I tell my old self and my younger self, just continue to have fun. We're blessed to be able to do this job and to be truly creative, relatively independent, and ask questions that if answered, ultimately really advance human health and advance human knowledge. It's a very privileged place to be. And sometimes we get bogged down. We see things that happen, way above us, above our control, that seem, sometimes haywire and chaotic at best. But, and while that can be discouraging, my advice to my younger self and my current self is that Remember, this is supposed to be fun. It is a privileged position to be able to wonder and discover and share that information with the rest of humanity and make humanity hopefully a better place. And so, just keep the eye on the prize. We'll get through some of the trying times, and again, there will be daylight, and we'll still be in a privileged position at the end of it.
Well, I'm inspired. That was amazing. Thank you. And I don't think that your younger self or your older self would be appreciative of. I think that many people who hear this conversation would be very appreciative of that information. And this goes across every industry that people are in. So thank you so much for that, Dr. DeBosch. And I know I said that I had one last question, but now I have one last question. What inspired you to get into this work? Was there a personal connection to this work, or were you just tapped into your wonderment and, just being inspired by research and all of that, what that entails? What inspired you to do this work?
It's multifactorial. Okay, so for example, when I was 5, I got a really bad rash on my skin because my sister and I, playing in the backyard, ground up a bunch of different leaves and grasses and dirts and rocks and things like that into a potion that we thought would make us invisible. We rubbed it all over my skin and I got a massive anaphylactic rash. No, it wasn't anaphylaxis, but a pretty bad rash from it. And that was the beginning of my scientific career. But put it this way, what got me into this line of work? It was at the most proximal point of attack. It was my mentors. As an undergraduate, I just wanted a job to make money as a dishwasher in a laboratory. I just wanted pocket money, and so I applied to a laboratory, and I met a literal a literal hero, but also sort of a figurative hero. Meaning, his name was Hirohito. He was a postdoc from Japan. He introduced me to the world of physician scientists. I could not believe that this man treated patients in the clinic and did really, really awesome science at the bench in the laboratory. And he was a true hero. He inspired me to do the same, and I did. And I never looked back, and I don't regret it one iota. Not one bit. I'm doing the dream job. LeBron James gets paid to dribble a basketball. I get paid to hold a pipette. It's all the same. Well, he makes a little more, but that's okay.
That is just amazing. And both of y'all's jobs and work, it's completely different, but you're in your passion, and that's all that matters. I thank you, Dr. DeBosch, for this conversation, and that concludes our Behind the Lab today.
Wonderful. Thank you.